Hi, this video is intended to demonstrate how to set up and run the ARACNe component in geWorkbench. First, let's briefly touch base on what's ARACNe and what's geWorkbench. ARACNe is an algorithm that predicts potential functional associations among genes based on statistical analysis of gene expression data. It was developed by Andrea Califano's lab at Columbia University and has been demonstrated to be an effective tool in identifying transcriptional targets when combined with chemical validation and literature searches. Next slide illustrates the principle behind ARACNe analysis at a very high level. We know that when the expression level of transcription factor increases, the expression level of target genes it activates will increase, and the expression level of target genes it suppresses will decrease. ARACNe takes a large amount of gene expression data, at least more than 100 microarrays, as input, calculates the mutual information of each gene against all other genes to identify the pairs of genes that have statistically significant correlations. Those pairs of correlated genes have a very high probability to be the transcription factors and their correlated target. In a more complex scenario, one transcription factor could activate the expression of a second transcription factor, and in turn, it activates a target gene. The mutual information calculation will result in the prediction of 3 correlated pairs, two direct correlations and 1 indirect correlation. The way that ARACNe handles the situation is to identify and eliminate the weaker correlation among the 3, therefore increase the probability of picking up only the direct correlated gene pairs. This process is called a data processing inequality. To have a better understanding of ARACNe algorithm and how to design ARACNe analysis, it's highly recommended to read original publications. The links are shown here. So, in summary, what ARACNe does is that it takes a large amount of gene expression data, at least the size of more than 100 arrays, as input, calculate the mutual information of each gene against all others to identify statistically significant correlations among genes. It also uses data processing inequality methods to further eliminate the indirect correlations. The output of ARACNe analysis is a predicted gene induction network, as depicted in this diagram. Now, what's geWorkbench? geWorkbench is a software that integrates many genomics data analytical tools into one easy access desktop application. It's free. At present, there are more than 70 available plugins supporting visualization and analysis of gene expression data, molecular interactions, DNA and protein sequences, and protein structures. You can find out more information about geWorkbench, such as the kind of analysis it performs, the input data format, and how to download and install the application at the geWorkbench's main project site. The CA Knowledge Center site is also a good source of reference. ARACNe is one of the components that has been incorporated in geWorkbench. There are more information about ARACNe component in geWorkbench on this website. So as mentioned at the beginning, this video is focused on demonstrating how to set up and run ARACNe in geWorkbench. We will use a small set of sample data and take you through how to load data, set parameters, run analysis, and view the result.